not an expert on these things, but I thought I'd remind you. So the first speaker today is actually from Los Angeles, right? You work here?
Yes.
How long have you worked here?
1.5 years.
Okay, so
Yeah.
very fresh,
Yeah.
very young faculty here at UCLA. The title of the talk is Large-Scale Genetic Studies of Human Complex Traits, and Jae Hoon is going to be presenting his first slide. Whenever you feel ready, start.
Okay.
Now
Yeah,
we're
yeah.
right on time.
Yeah, I can just start. Yeah, thank you for inviting and thank you for your great introduction. So, I'm Jae Hoon. So I'm a faculty in the Department of Psychiatry and Behavioral Sciences, and I'm also affiliated with bioinformatics and genetic genomics. Okay, I will try to speak up, but yeah. Okay, can you hear me now? Okay. So, yeah. So before introducing myself, I have a few slides that I want to start with that describe the background of my research and then the types of research that I'm doing. And I guess some of the UCLA people have seen these slides, so sorry for the duplicate slides. So the goal of the genetic study is to find the genetic basis of human traits, which is to find the genes or genetic variants that influence the trait. So to achieve this goal, genetic studies have used both linkage and association studies. So in linkage study, we analyze families, and in association study, we have cases and controls. So in both approaches, we need to know the genetic variants of these individuals. And recent development in microarray or whole genome sequencing enable cost-effective approach to genotype these individuals. So for example, this Omni 2.5 million microarray can identify about 2.5 million common SNPs present in human genome, and this high coverage whole genome sequencing can identify millions of rare variants as well as common variants. And these technologies have changed the paradigm of genetic studies dramatically. So for example, this early linkage study for schizophrenia about 20 years ago analyzed 35 affected individuals in 9 families and about 300 markers. The latest GWAS for schizophrenia analyzed about 37,000 cases and more than 100,000 controls. So this is much, much larger. And then there's been large GWAS like this for the past few years. And over the past few years, the cost of genome sequencing has decreased dramatically, as shown in this figure. And I guess many people have seen this figure. And genetic studies are moving toward genome sequencing from microarray. And there are a few large-scale genome sequencing projects, so such as whole genome sequencing in psychiatric disorder, WGSPD consortium. And there's also and then genome sequencing project, GSP, funded by NHGRI, and there's also TopMed project, some funded by NHLBI. So these programs are trying to sequence at least tens of thousands individuals or sometimes 100,000 individuals. And here at ICN and UCLA, we have already generated whole genome sequencing of few hundred or even thousands of individuals, and we are analyzing this large-scale genetic data. And genetic studies are facing two major challenges in handling this large-scale genetic data set. So first challenge is developing methods that can handle this large genetic data set, because methods used to work great for small sample size may no longer work for large sample size. And one of the examples is a linear mixed model, which is a method to correct for population structure in GWAS or estimate heritability. So original linear mixed model was able to handle only a few hundred individuals. So it was mainly used for mouse or other model organism study. And the problem with that was that it estimates the variance component for every variant. So I developed efficient linear mixed model called MIX, and it can handle up to 10,000 individuals. And another challenge is actual analysis of the large genetic data, because the procedures to analyze this data have become increasingly complicated and have several challenges. For example, these are the typical procedures to analyze the sequence data. So first, we need to call the variance, and the challenge here is the amount of computational power and storage, because we need to handle terabyte or sometimes petabytes of data. And then we need to perform quality control, whose goal is to ensure the quality of sequencing data, which has a lot of noises. And then we perform association analysis to find the variants that affect the traits, and we can do several other analyses. So my research goal has been addressing these two challenges. So specifically, our goal of our lab is to develop techniques that will enable understanding of especially neuropsychiatric disorders utilizing this large amount of genetic data. So I've been working as both method developer and then analyst. And since I joined UCLA, I've been focused more on the analysis part. And I've been involved in several whole genome sequencing projects. And because this is CGSI, I'll focus my talk on the method development. And I hope to give you I'll talk about 2 research projects, but let's see how time goes. So first, I'm going to discuss the rare variant testing for large families. So as we all know, GWAS have identified many common variants associated with many common traits, but we also found that they explain only a small fraction of heritability. And there can be many other potential contributors to this missing heritability, such as rare variants, structural variants, or gene by environment interaction. And recently, rare variants have drew great attention because we found that they also affect complex traits, and they are known to have large effect sizes. However, because it is statistically very difficult to detect effect of single rare variant, the collapsing approach that combines information of multiple rare variants in one gene has been proposed. So this approach is trying to ask whether group of rare variants in one gene influence a trait or disease. And there's been numerous methods developed for detecting association of rare variants, and some of them are shown here. And I also developed like 3 different methods, but very few people use it. Okay. And several genetic studies have applied sequencing, have performed sequencing and applied this method to identify rare variants, but they have not been very successfully identifying these low-value genes. So for example, this top paper did a whole exome chip analysis on more than 50,000 individuals, but they didn't find any novel gene using CMC or SCAT. The bottom paper did the whole exome sequencing of more than 3,000 individuals, and they did a replication and follow with more individuals, but they found only 1 novel gene using the CMC and SCAT. Yeah.
These papers using what you had on the previous slide?
Yeah.
Methods that actually
they are very, yeah. So the CMC and SCAT. So CMC is this paper, and SCAT is this paper.
Okay.
Yeah. Yes, okay, yeah. And this is not, however, very surprising because Shamir and his colleagues show that thousands of individuals are required to reach the acceptable statistical power to detect effects of rare variants associated with a trait or using exome sequencing. Then can we do something different? Because I mean we have to wait until the sequencing cost drops. So another method is family-based studies. So one advantage of family-based studies over case control is that the rare causal allele may be enriched in some large families, which increase power to detect their effect. So this is a figure from paper by Zhu and Jiong, who show that families indeed have higher power to detect effect of rare variants compared to case control. So here red line is the power of the 3-generation family, and the blue line is the unrelated individuals. And there are several other advantages of family-based studies. So one of them is that we can detect and even correct the erroneous scores made by next-generation sequencing by considering by considering Mendelian inheritance. So there are several tools like Color, Polymute, or CGSI that can detect these Mendelian errors. Yeah.
When you speak about family studies, you mean trio studies?
I mean, I'm talking more about like extended family for this case. Before like extended families, more than trios, right? Yeah.
So not necessarily trio. It could be
Any structure, yes. Any structure, yes. Yeah. Yeah. Just let me know if you have any question, okay? And then another advantage is that we can detect de novo mutations and then study their effect. And also, family-based studies can be designed to be robust population structure, which may cause spurious associations in case control studies. And the last, or not the least, one of the important things is that these genetic studies have already collected these families from linkage studies. So they are already available for sequencing. And then TOMMAT is doing the sequencing a lot of families. So, and also they are most likely well phenotyped as well. So we have a lot of phenotype information. So we need to utilize these existing resources. So these are the reasons why family studies drew great attention recently, and there are many methods developed for family-based studies. But some can handle only trios or nuclear families, like small families that you mentioned. And some can handle only quantitative traits, and these are mostly based on SCAT method. And some even don't provide software, so I couldn't even compare their methods. So there are very few methods that can handle these large pedigrees, both binary and quantitative traits, and affected only pedigrees. And also, I'm going to show later, but they don't usually handle missing founders correctly.
Affected only pedigrees means that you don't have control data.
No, yes, that's correct. So because in the linkage study, for linkage analysis, you only need affected. So for some people, they only call it affected individuals from pedigrees. But for the association, like previous approaches, they usually do association by taking into account the family structure. But then they need the controls. So those methods cannot be applied to affect only pedigrees. Yeah. So this motivated me to develop method called rare IBD. So the main idea of this approach, this method, is that causal rare alleles are enriched among affected while they are deflated among unaffected. So I will first give the high-level description of my method. So first, I assume that only one founder has a mutation in each family for each rare variant. This is most likely true for most rare variants because it's very unlikely that two founders in the same family carry the same rare mutation. Then any non-founders who have this mutation share this allele IBD. So we are interested in segregation of this allele, which simplifies IBD estimation. We then compute a statistic that measures the enrichment of this allele among affected and depletion in the unaffected. And then we then take a burden approach that aggregates statistics across multiple rare variants and multiple families. And we are trying to find whether the rare variants in a gene correlate with the disease status. So here are some examples that show what my method is trying to do. So in this family, there are 6 affected and 6 unaffected. And 3 affected have mutations, while 3 unaffected have mutation. So this variant is unlikely to be causal variant. But for this one, there are 5 affected have a mutation and only 1 unaffected have mutation. So this may be causal variant. So my method is trying to find the correlation between the genetic variant and then the family structure. So here's a formal description of my method. So first, I look for variants where only one founder has a mutation in each family. And for such a variant R in family J, we define our statistic Sij as sum of the number of affected with mutation and the number of unaffected without mutation. And we are then interested in the value of this Sij on the uniform distribution of what's called inheritance vectors. So to estimate this, we enumerate all possible inheritance vectors and estimate the mean and standard deviation of the statistics on the uniform distribution. So this is the mu k and sigma k here. And then we can estimate the g-score of our statistic. And then this is for one variant and in family J. And then we can take the weighted sum of g-scores across all rare variants. There are m rare variants and all families n families. And then we calculate the g-score for this gene. And to evaluate the p-value, we can estimate the p-value from normal distribution or by gene dropping, which I'll discuss later. I mentioned this is 1F because we are estimating the g-score for only one founder that has a mutation. And I'm going to relax this assumption later.
So
Yeah.
K is the index for the founder?
No, there's a founder who has a mutation, who has a rare allele.
One founder. Okay.
Yeah.
I was trying to understand what's the K that indexes the mu and the sigma.
Oh, this is the mean and standard deviation of this statistic, assuming that this founder has the mutation.
Okay.
Yeah.
And it's the mean and the standard deviation across all the family?
All the family, or in this family, in one family. Yeah,
Okay.
yeah. So you can think that we have to estimate this for each founder in each family. Yeah.
And what's this uniform distribution thing?
So this is like a null distribution. You can think of like null distribution. So in the family, if you enumerate all the possible inheritance vectors, meaning that then you are getting the uniform distribution of the statistics. Yeah. This is how the non-parametric linkage studies analysis usually work. Yeah.
And the
Yeah.
aggregation is just done by adding the silver mutations within the same gene.
Yeah, same gene. Yes. Yeah. Yeah. Any question?
What are the weights that you use for the family?
Huh?
I see the W, the weights.
Oh yeah, weight. So I'm going to discuss later. Yeah, yeah, that's a good point. Yeah, so we take weight and then I will show you how we estimate the weight. Okay. Okay. Can I go? Yeah. So there are a few statistical and computational challenges in this method. So I will go over them one by one. So one problem is that the founders are usually missing in these large families because we don't have their DNA, right? Because we cannot even get their DNA because they are not here, right? So which can cause two problems in our method. So first problem is that we may not know whether only one founder carries a mutation. It is possible that two or more founders can carry the mutation. So to solve this problem, we can filter variance according to the minor allele frequency of other families, using other families, or reference populations such as one-ton genomes or exec. The second problem, which is more difficult problem, is that we may not know which founder carries a mutation. So to solve this problem, we decide to estimate mu and sigma for every founder in the family, and then we add them and then we divide them. Basically, we average the scores across all founders. So here, Fj is the number of founders in family J. So we calculate the G-score for every founder from K for all founders, and we divide by Fj, and this is all F because we are using all founders. And then we calculate the G-score for gene using this statistic. And the second challenge is that enumerating all inheritance vectors takes too much time because the number of IV is exponential in the number of non-founders. So for n non-founders, the enumerating all IVs takes 2 to the 2 times n. So if number of non-founders 40 is too huge. But since we are only interested in mean and standard deviation, we can just do the random IV sampling, just fixed number of IV, like 100,000. And we found that the mean and standard deviation do not change much even if we do more than 100,000 inheritance vector sampling. And the third challenge is that getting the p-value from normal distribution is not very accurate. So here, this is the first positive rate of my method using 3 different pedigree structures I'll show you later. But at alpha equal to 0.05, the FPR is much lower than, not much, but it's lower than 0.05, which means that we are losing some power. So the normal approximation tends to yield the conservative p-values. And then later I will show we also estimate weights from the data. Then the weighted sum of G-score is no longer normal, asymptotically normal. So we cannot use normal approximation anyway. So the solution is we do the gene dropping to estimate the p-value. Yeah.
What's the rationale behind your false positive being always lower than what you expect? Do you overcount things, or what's the conclusion?
I think that my distribution of G statistics are not actually normal. It's the standard deviation is not 1, I guess. It's much lower, yeah. Yeah, I think that's what people found with the linkage study as well. Yeah. Yeah.
Why would you expect your false positive rate to be directly related to your p-value in your alpha? Like I feel like alpha and false positive rate are measuring 2 different things.
Oh, sorry, so this is the threshold, the 0.05 threshold? Sorry, so this is the, if it's 0.05, then your false positive rate should also be 0.05.
Oh,
Yeah.
it's not false discovery.
Yeah,
it's
sorry,
only
yeah,
false
yeah,
discovery.
yeah, yeah, it's okay, yeah.
So this is false positive is divided by the null. It's out of the null things, not out of the discoveries.
Yeah,
Yeah, sorry.
yeah, it's okay, yeah. So this is how the traditional gene dropping works in the family studies. So we have this family. So we first generate the founder genotypes according to the population frequency. And then genes are dropped to non-founders according to Mendel's law randomly. And then we compute the statistic of interest. In this case, it's Sij. And then we need to enumerate IBs again to estimate the mean and standard deviation. And then we compute the G-score. And we repeat this for all families and compute the sum of G-scores. And then we have to repeat these steps many times to estimate the p-value. So this is kind of similar to permutation test in case control studies to simulate the null distribution. The problem with this approach is that this step is computationally very expensive because we don't want to enumerate all IBs again for every gene dropping replication. So can you do this fast? So there are two observations from our approach. The first is that since we assume that only one founder has a mutation, the number of possible genotypes is kind of limited. In other words, there can be only F possible genotypes, where F is the number of founders, because each founder, because we are only assuming that only one founder has a mutation, and there can be only F founders, there can be only F possible founder genotypes. And then for each of F founder genotypes, the mean and standard deviation of SIJ statistic is only fixed. It's fixed. We only need to estimate only once, because the mean and standard deviation does not change how genes are dropped to non-founders. So what we do is that we pre-compute this statistic for each founder, and then we assign the mutations to one of the founder randomly, and then we use pre-computing mean and standard deviation, which makes this gene dropping very fast. Okay. And then the last challenge is the one that you mentioned. So what is the optimal weight? So nobody knows what the optimal weight, but we know that it is very important to upweight the causal variance and downweight the non-causal variance to increase the power. And people came up with two different approaches to calculate the weight. So first is kind of frequency-based weight. So basically, if the variance have minor frequency less than certain threshold, then we think they are causal. So CMC use the fixed threshold like 1%, and we use the variable threshold approach developed by Price et al. And then I'm going to discuss more of this later. And then the another approach is effect size based weight. So basically, we estimate the effect size, and then we weight each variant by estimate effect size. And we use ERDC approach by Ling and Tang. Yeah.
Question about the founders. So if you don't know the genotypes of the founders,
Mm-hmm.
I guess it depends on how strongly can you assume that they're the real founders. So there might be like
Huh?
might be some close inbreeding, right, in the earlier ancestry that you don't know.
Oh yeah, yeah.
Any way to try to capture that?
So you mean that there can be more than one founder with a
There could be
mutation, right? Yeah.
One founder that you just don't know is deeper in the tree, and it might be the cause that there's actually
So in our method, we don't really care which founder has a mutation. We just because we are calculating the G-score using all.
Yeah, but I guess what I'm
Yeah.
saying,
Yeah.
if there is
Hmm?
a deeper part of your
Oh,
a deeper part of your genealogy that
you are not aware of.
for that
Yeah.
has like
Yeah.
close
Mhm.
relation.
Yeah, I mean,
Is there a way at least to maybe try to assess the probability of that there's just more excess than you'd expect from a single founder in this
mhm.
in the genealogy that you have in the family that you have?
So I guess that if you know more family structure, it's better, right?
Yeah.
I mean, but if you don't know, then we just have to assume that the mutation came from somewhere, right? Somewhere in the tree. But
It
only
could
one.
have come from
Huh.
somewhere that you don't know,
Mhm.
and therefore, at like the founders that you do know, there might be multiple founders that have that mutation.
Yeah, 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 yeah.
Because they're actually
Yeah.
related in
Yeah.
some way. And you can, I guess, try to.
Could you generalize the method
Yeah.
dropping this assumption that it
Yeah.
must be one of the
Yeah,
founders
yeah.
of
I
that
mean,
mutation?
Yeah, I mean, I try to drop that assumption, but it's very hard. So, but
At least
yeah.
maybe try to
Yeah.
assess the fit, like the fit to that of that assumption.
No, actually, so I'm going to show you later, but I'm going to show you that when the more than one founder has a mutation, then our method has a little bit of inflation, right? But it's not much. So, yeah, I'm going to show you later.
So
Yeah.
you think it's
Yeah.
actually robust to this type of thing? That
Yeah, unless it happens really a lot of times, but if it happens a little bit of time, then it's fine.
Okay.
Yeah. But I mean, you're right. I mean, then we are because our method is ignoring this case, right? So I mean, we may lose power, right? Because this may be a causal variance, right? So, but yeah, because it's really hard if we try to incorporate two founder having mutation.
And I'd say
Yeah.
maybe you cannot do that, but at least try to see if the case if the family trees that you have
Mhm.
just does not support that assumption for some reason, or
I mean, it's not some. I mean, it depends on the earlier frequency variance, right? If it's really rare, then it's very unlikely that two founders. I mean, even
unless they're
yeah,
related,
in the
that's the
inbreeding, yeah,
problem, then,
yeah,
that-
yeah.
and a lot
Yeah.
of these families, it's
Mhm.
possibly more likely than in a general family. You have that.
Maybe,
That might
yeah,
be the cause,
yeah,
such
maybe,
as
yeah,
that you actually see this phenotype.
yeah. That's true. Yeah, 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 yeah.
Are you assuming it's rare in the population or rare in the family?
Rarer in the population, right? But the founders should be unrelated, right? So they should. I mean, unless there's inbreeding, right? They should be unrelated. So they should represent the population minor frequency, right? But my point is that it should be common in the family to detect its effect, right?
Which is why I
Yeah.
wanted using the family study.
Yeah, yeah, yeah. So wait. So this is the various approach for AIBD. So for the fixed threshold approach, the weight is 0 if the minor frequency is greater than a certain threshold, let's say 1%. So for the VT approach, it varies this threshold and try to find the maximum Z-scores. So let's say T is the minor allele count among founders in all families. And for T from 1 to max allele count, we compute this Z-score. So weight is 1 if the minor allele count of this variant is less than or equal to T, or 0 if it's greater than T. And then we compute this Z-score for T equal 1, T equal 2, up to T equal to maximum allele count. And then we find the maximum Z-score among these values. An important thing is that we have to do this in the gene dropping as well, because we have to do the same thing in the permutation as well. And this is the EREC approach. So basically, we estimate like odds ratio, and then we add some constant. That's what this approach does. So basically, our weight is no longer 1, but it's log odds ratio plus 2 if the minor allele count is less than or equal to 2, or 0 if greater than T. And we find the maximum Z-score. So to test our method, we first generate simulations to estimate the false positive rate and then power. And here's how I generate the simulation. So first, I need to generate the haplotypes of founders. Haplotypes of founders. And to do that, we generate the haplotypes unrelated individuals in coding region using what's called COGI software, assuming the EU population. And then we assign one of the rare mutations to one founder in each family. And then we generate the haplotypes of non-founders, assuming some inheritance vectors. And then we use 3 different pedigree structures, which I will show you later. And then given the haplotypes, we need to assign the disease status to each individual, and which I'm going to show you in the next slide. And then we can estimate the FPR and power of our method and other approaches. So this is how I determine whether individual is affected or unaffected. So let PI equal to 1 be the probability that individual A is affected. So in the first positive rate simulation, they are basically 50%. It doesn't depend on any genotype. It's just random, right? But in power simulation, it is based on logistic regression, which depends on several parameters, such as baseline prevalence, which I assume 40%, and odds ratio, which I assume to be 2 for regardless of minor frequency, and X is the genotype vector.
So is that 40% think the number? Are you actually thinking of diseases with a prevalence?
No, this is not very good term. This is not actually prevalence. I mean, this is like basically the probability of having a disease, having all other genes except this gene. So you are trying to simulate one gene, right?
I see.
Yeah. So
Okay.
you have all the
It's
other
not the
genes.
prevalence
Yes,
of the
but it's
disease
like
in the population, it's kind of
yeah. So this is not actually
importance
prevalence.
of
Yeah,
that one
yeah,
gene that-
yeah, yeah,
okay.
yeah. If I make the prevalence too low, then I cannot really simulate anything. Yeah. So yeah, this is not very good term, but I don't know how to define this term. So,
Okay, as
yeah,
long as
yeah,
you understand,
yeah,
that's great,
yeah,
thanks.
yeah. So and then we also change the CI value, where CI is the probability that the rare variant is causal. So not all the variants are causal, so we change the probability of the variant being causal. And then we also limit the number of affected individuals. So minimum number of affected individuals is 1/3, and the maximum is 2/3, because we rarely see the families with all affected or all unaffected. And then we also simulate the missing founder case. So in this case, we assume that top 2 generations are completely missing. So this is the pedigree structures that I used. So there are 3 different pedigree structures. The first one is wide. There are 30 members in the family, and this kind of wide, right? And then this another is deep, where there are 36 members and 20 such families. This 4 generation. And we also simulate a small family where there are 12 members. So this is not actually very small, but compared to others, it's small. Yeah. So yeah. So we try to see how our method works in the other small cases, in the small families. Yeah. So first, I want to show the effect of the weighting the variance and then the OLF versus 1F approach. So I have 4 different methods. So weighted OLF means that we use the mean and standard deviation of all founders and average the G-scores. And then we also use the frequency and the effect size based weights. And weighted 1F means that we use the same weights, but we only use the mean and standard deviation of only one founder for some mutation. And unweighted 1F means that we don't have any weight, and affected only means that we only use affected individual in the statistics. And I didn't show here, but they all have correct false positive rate. And these are the 3 pedigree structures. And as you can see, by weighting the variant, so this is red versus the green, so the weight increases the power significantly. And then by using the OLF, we can increase power further. So we decide to use the weighted OLF for the rest of the simulation and all the approaches, all the analysis. And this is the first positive rate between our method and other approaches. So this is where all founders are genotyped, and this is when the 2 generations are missing, top 2 generations. And this is my method, and this is another method called FPCA, developed by Zhu and Zhirong. They have 5 different methods. And I also test another method called PEDGene, which has corner, which is like SCAP, and burden approach as well. So when all founders are genotyped, my method is a correct FPR, and then the burden approach of PEDGene also has correct FPR. And for some of them, they have very high FPR. So I did not include them in the later analysis. And where top 2 generations are missing, our method still has correct FPR, but the burden approaches, they have a slightly higher false positive rate than they expected. Right? And then this is the power between my approach and other methods. So this column is all founders are genotyped, and this is when top 2 generation missing. And this shows that our method is more powerful than all other approaches. And the most important thing about this plot is that when the top 2 generation missing, our power does not decrease a lot for the wide and deep families. It is because we use the information how the causal alleles segregate among non-founders, but the top 2 generations do not provide much information about those segregation. Yeah.
I was also kind of noticing that the performance of your approach and the burden also kind of stay the same regardless of the type of families, like wide, deep, small. They kind of perform in the same ballpark, while some other ones of the approaches seem to change their performance more depending on the
that's true.
structure that underlies the data.
Yeah, that's true. That's a good point. Yes. Yeah. So for other approaches like burden approaches, their power depends on number of overall individuals. So by excluding the top 2 generations, they lose much power. But we use segregation information.
And it's just
Y-axis is same between these 2. Y-axis same between these 2. Between these 2. Yeah. So you can just directly compare. Yeah. And then also, as I mentioned, so one of our assumption is that only one founder carries a mutation. So we simulate case where there is 30% probability that 2 founders carry the same rare variant. And then in this case, our top 2 generation missing. And we can see that our method has slightly high inflation, but it's not much. So wide, deep is okay, but small, we have a little bit high inflation. But compared to other methods, this burden approach, they have much more high inflation. So I don't know why they don't even assume that top founder only one founder has a mutation, but still they have high FPR in this simulation. And then also some people like the idea of having the protective rare variant. So I simulate a case where 40% of rare variants are protective with odds ratio of 0.25, and 60% of them are deleterious of odds ratio of 4. And then we also assume the top 2 generations missing in this case. So in this, when there's protective rare variants, we know that the sketch-based method, so kernel approaches, they have high power. So as you can see, the kernel approach has higher power than burden approach for the PET gene. But our method is still more powerful than kernel approach. So now we decide to apply this method to 2 real family data sets. So first data set is early onset chronic pulmonary disease, EOCOPD. There are about 347 individuals in 49 extended families. This is high coverage for exome sequencing, and about 100,000 variants after QC. And there are about 12,000 genes with at least 3 rare variants. And we define rare using the allele frequency from families and the allele frequency from external sources such as ESP, DB, 1000 genomes. And there are about 155 affected, 148 unaffected. Yeah.
What does early onset mean for COPD? I haven't heard of this. So these people get sick how early in life?
I'm sorry, I don't know. yeah. I'm not very familiar with this disease either.
Right,
Yes,
so it
yeah,
was
sorry.
just a
Yeah,
curiosity,
yeah,
this
yeah.
is something
Yeah.
that usually happens late in life, and
Yeah.
I wanted to have a
Yeah.
okay.
Yeah. And then the second data set is what's called Cleveland Family Study, CFS. So here we measure the sleep. So these are the individuals with sleep apnea in both African American and European American, so AA and EA. There are 563 AA and 665 EA in this number of families. And this is not sequencing, this is SNP chip with exome chip. So we have a lot of SNPs. But because this exome chip does not capture all of the rare variants, the number of genes with the rare variant is smaller than this. So we only have 7,000 or 6,000 genes with at least 3 rare variants with the allele frequency from families or from external sources. And this is number of affected or unaffected individuals. And we analyzed EA and AA separately. So this is the result of our approach on the EOCOPD data set. The left plot is the QQ plot, and it shows that p-values are well calibrated, and the lambda GC is about 0.8 to 0.83. So it's a little deflated, but it's not inflated. And this is the top 10 genes, and this is the Manhattan plot. And unfortunately, none of them have the genome-wide significant p-value. But we also applied other methods such as FPCA and PET gene to this data set. So this is chi-scale form, which is this is so I'm going forward. Should go backward. So this is the chi-scale form, which has slight inflation. The lambda this is 1.5, I guess, and then CMC is kind of weirdly they have deflation, very severe deflation. Lambda this is 0.2, and then for the PET gene approaches, they both have a little bit of inflation, but this part of the p-value is kind of weirdly deflated. And then I apply my method to Cleveland family African American data set. So this is QQ plus, so lambda GC is 1.04, and these are top 10 genes, and we almost have the genome-wide hits, but it's not significant. So, and this is the CFS European American data set, and kind of same, but we don't have a very significant genes. And we apply the FPCA and other approaches to CFS EA data set, and the results are kind of similar. They either have very severe deflation or inflation. So lastly, I want to discuss how my method is robust population structure. So there can be 2 sources of population structure in the family studies. The first is the within family population structure. So this means that some of the founders are from different populations than other founders, and they may have many more rare variants. And the problem with this is that, so basically I didn't mention this, but the assumption in our gene dropping approach is that the rare variants are independent. But the problem is that if one founder has many rare variants and it gets transmitted to multiple non-founders, then it creates what's called perfect LD. I mean, they are just duplicate variants. So they are not independent anymore. So to simply solve this problem, we remove the variants that are in perfect LD with another variant in the family because they don't provide additional information about the segregation of the variants. And we did a simulation where one founder has 4 times more rare variants than the other founders, and our false positive rate is a little bit inflated, but it's not much, about 0.05, 0.57, 0.0574. And another source of population structure is between family population structure. So it is basically families with different ancestries. And our method is also robust to this kind of population structure because you can think of my method as the meta-analysis across many families. Basically, we are doing the various sum of z-scores, which is similar to the fixed effect model of meta-analysis. So to show whether this is okay, we merge the CFS AA and EA data set and we analyze it together, and then we still have correct distribution of p-values. So lambda GC is not very great. It's not very high. So in summary, so we developed a rare variant association method called rare IBD for large families, and we use an insight that only one founder carries a mutation for rare variant, and we show that our method is more powerful than other approaches in simulations, and it generates the correct p-values in the 2 real family data sets. And its another advantage is they can correctly handle missing founders, and it is robust population structure. And I didn't show here, but it can be easily extended to affecting only and quantitative phenotypes. And one caveat using my method is that all the missing genotypes need to be imputed, and it's not hard for this to meet this requirement because there are several tools like Polymer or ShapeIt. They can impute your family data set very well. And my software is here, and then this was published in American Journal in last year. Yeah. Do you guys have any idea? So I think that because of time, I don't think I will have time to go over other methods. It's based on the EQTL method, so I'm going to skip it. And then, so I'm just going to acknowledge all the people. So this was work that I did when I was in Harvard Medical School. So my PI was Shamil Snayab. And this is actually a picture of my lab. This is after having Korean barbecue. So we eat a lot, or we work a lot too. So yeah, thank you very much. Yeah. Yeah. Yeah.
June, I was wondering if you thought about aggregating across, let's say, gene sets or pathways, and
Yeah.
the methods would work to increase power, because it seems like your method is more robust than the other.
Yeah, I mean,
I think the sample size.
yeah, people are trying to do that, but it's very difficult to find the pathway, right? Right pathway for the certain disease, right? So I mean, yeah, I mean, we can easily do that. We can just that means we just having more genes, right?
Yeah.
So so I mean, we are open to do that, but still we are having some trouble, right? Finding the right pathway, especially for certain disease where you don't even know which genes cause your disease, right?
Yeah.
Yeah. But yeah, people are doing that.
But you haven't tried like just a general like go gene sets or tissues or what people are using just to aggregate and see if you
No.
say you have a brain phenotype or something and
Yeah.
central nervous.
No, I haven't tried that. Yeah.
Following up on this, and I don't know if it's related or not, but you may be studying something like, I don't know, early onset COPD. Maybe it's not known what pathways are relevant for this.
Yeah.
But there may be pathways that are known to be relevant for cousin diseases, diseases that are kind of similar.
Yeah.
So there may be some information to try.
I agree. Yeah, I agree. Yeah. And then the important thing about the pathways, actually, one gene may increase your heart level, when the other gene decrease it. So if you just naively merge them, you won't get any power. So you have to know this direction. So it's kind of hard problem too. Yeah.
No more questions? Okay, let's thank you.
Okay. Thank you.